All right, so this handout should look a little familiar. I showed you the basic form of this at the very beginning, and then now we know more detail. So you know about 80, 90% of this already. We just need to add some details. So what can we add to this, assuming going left to right?
Light will be on the yellow.
Totally agree. Yep. Light. What else? Water.
Oxygen.
Oxygen. I agree. 3 different things can go here.
Photosystem 2 and photosystem 1.
Photosystem 2. Photosystem 1, which are both what kind of reaction?
Dependent.
Light dependent. I agree. The products of the light dependent reaction are ATP, NADPH. That H right there is really important because in that hydrogen right there are the high energy electrons that were excited by photosystem 1. Cool. What's all going on over here?
The Kelvin cycle.
Kelvin cycle. Which is light
Independent.
independent. What else? I'm sorry?
That's
CO2, I agree.
and then glucose at the bottom.
I agree. You know more than that.
G3P.
Yep, G3P.
Yep.
Which of course then it's the glyceraldehyde part of this molecule that contains the high energy electrons. Cool. What can go right there? At about 10 o'clock.
RUBP.
Completely agree. RUBP, what will go there? At about 2 o'clock. EGA, which is then converted to BPG. Totally. Now, don't forget, right in here, there's oxidation and reduction, right? Your NADPH is oxidized. It gives up its high energy electrons. The BPG is reduced into G3P. Okay. Now, G3P, don't forget that it makes 6 of them, right? So 6 of the G3Ps are here. 1 of the G3P leaves, and then G3P actually returns here, is converted into triose phosphate, TP, whatever, right? And then TP is converted back into RUBP. Now, do you have to worry about all of the intricacies of the carbon backbone and how the oxygen groups are shifted around? Absolutely not. Okay. The substrates here, very, very, very few people call them by their whole name. We all use RUBP, PGA, BPG, G3P, of course. Okay. Now, an important thing to understand is that that should be a 3. Bummer. G3P. This can also go to the mitochondrion for the production of ATP. So G3P sometimes is used for the production of macromolecules. Or the G3P goes to the mitochondrion for the production of ATP by cellular respiration. Okay, so it depends on what the plant needs. If the plant needs energy, then the high energy electrons through cellular respiration are used to make ATP, or if it needs to make macromolecules, that happens as well. Okay. It's a really, really cool summary graphic. Any questions on this before we leave it? I'll post all of these. You okay? All right. So then there is some graphs. Hey, this is group 4. Check it out. So what are we looking at here? Okay, what we're looking at here is the electromagnetic spectrum. Okay, back in core 2, core 3, right? We talked about the electromagnetic spectrum all the way from, gamma rays, which are really, really compressed, to radio waves, which are really, really stretched out. That's all the electromagnetic spectrum. Okay. Whereas this is a tiny, tiny slice of the electromagnetic spectrum. We see very little of what actually is out here. Okay, there's ultraviolet rays out here, there's infrared, there's radio waves, microwaves, gamma rays. There's tons of energy flowing through the atmosphere, and we don't see it. We see the small slice that we refer to as visible light. So these are wavelengths of light, technically in nanometers, if you want to know, right? So we should drop some links in here because they're important. This goes from 700 nanometers, right? And this goes down to 400 nanometers. So this is 4, this is 5, and this is 6. So the different colors of light that we see are different wavelengths, okay? So these are wavelengths of light, the classic red, orange, yellow, green, blue, indigo, and violet. Roy G. Biv. Fine. Okay. Well, what is this? What is the y-axis? The y-axis is the absorption. This is the absorption of light by pigments in the chloroplast. That chloroplast is that green bag inside of all photosynthetic organisms, and they contain pigments. Pigments absorb light, right? Well, what do the pigments do with the light they absorb?
Use it within photosynthesis.
Use that energy within the light-dependent reactions and excite electrons. So the more energy that a plant can absorb, the more electrons the plant can excite, the more G3P the plant can make, therefore either the more ATP or the more macromolecules that the plant can then produce. Okay, so plants don't have just one pigment. All right, the more pigments they have, the better. Okay, so it actually turns out that there's 4 main pigments. Okay, there is a light green pigment called chlorophyll A. Light green? That's not right. Light green is B. It's the dark green that's A. Sorry about that. This is chlorophyll B. It is the dark one here. This is chlorophyll A here. That's chlorophyll A. Okay. And then these orange ones. You guys ever heard of carrots? Yep.
Yeah.
Carrots are, yeah, really, I hope so. Carrots are named after the pigment that they're loaded with. They're called carotenoids. That's why we call carrots carrots. Okay, because that orange pigment is a carotenoid. There's another group called xanthophylls, and those are your purples. Okay, so these different pigments absorb wavelengths of different forms. Okay, so over here, your dark, this is chloro A, and then your light, let's go, I like how it's slanted. Nope, let me change it. This is your chloro B. Where is there the lowest amount of light absorption?
Around green and yellow.
Around green and yellow, right? Why? Why is there almost no absorption here?
The wavelength
That
is
light
bounced
is
off.
reflected off.
Yep, the light bounces off. The light is reflected, okay? If you want to kill a plant, you shine green light on it. Always makes me giggle when you'll walk by, you're at the drugstore, you're at Walmart, you're at a 99-cent store, and they'll have this little stand of plant lights, and there'll be light bulbs that they've painted green. I look at that and go, that's the worst light to put on the plant. All right. You take a light bulb and you somehow color it green and expose the plant to green light. The plant absorbs none of it. You might as well take the plant and put it in a box. Okay. No light. All right. So every wavelength of light is good for a plant except for green, because of course, as you said, the green reflects, the green bounces off. That's why we see green. Okay. Don't forget that our vision doesn't come out of our eyes. People talk about looking at, and that's actually, it's the wrong way to visualize sight. How your eyes really work is our retina is nothing more than a satellite dish that receives energy. Okay. And so when we focus on different items, the light then comes to our eyes, and that's what we see. Fine. So we see plants as green because that's the wavelength of energy that is reflected, that bounces off of them. Right. Now, your blues, your indigos, and violets are better for the plants. The plants can absorb more energy. Red and yellow aren't bad. Okay. The plant will still gain energy from red and yellow, but really the blue and the violet. And if you go then a little bit more intense and you compress those wavelengths together just a little bit more, you then get ultraviolet, and plants really respond to that. They absorb tons of energy from what sits on this scale right about here, and that's what we call ultraviolet. Okay, fine. So if you were to take this information and create a graph that would outline the rate of photosynthesis, or the reaction rate of photosynthesis, what would the general graph look like? If this were reaction rate and these were the same wavelengths of energy, what would the reaction rate for photosynthesis, what would that graph look like?
It would look approximately the same, with most of the reactions happening on the left.
Yep. You get a higher peak here, it flattens out, and you get a smaller peak here, which is exactly what this is. Okay. All this is the same graph. Now, I would have liked if they'd taken this pretty axis and put it down here, but they didn't. So you go, all right, I can work with that. So here's 400 nanometers, and here's 500 nanometers, and here's 600 nanometers, here's 700 nanometers, fine. And these are wavelengths of light. Wavelengths of light. And of course, nanometers. And then here, this is your rate of photosynthesis, your reaction rate. And how do you measure that? You measure that measured by O2 production. Because as the plant goes through photolysis, releases oxygen, and so that is an indicator for the photolysis of water. So, classic question. Let's play the toddler. Why? Why is there a big hump here? Why is the reaction rate higher in our blue, indigo, and violet range? Why is that reaction rate higher?
More light is being absorbed.
More light is being absorbed. What does the plant do with that light?
Use it in light-dependent reactions.
Totally. That light is used in light-dependent reactions. That produces ATP and NADPH. Those two products are then used by the light-independent reactions to make G3P, right? So don't just say, oh, there's peak. Understand, always, conceptually, right? The vocabulary comes with time. Memorization is not the way to do science, right? It's concepts first, vocabulary always comes second. All righty, Miss Almasan, there you go. Any questions on that before we leave these two really cool pictures?
No.
Okay. Almasan's a quiet one. She just lurks in the background and listens. All right. So the other question that I asked that group to answer is why do we water plants? Even if you walk up to a child, right? You walk up to a little kid and you go, what do you have to do to a plant or it'll die? And the child will know, you got to water it. Everybody knows that. Why do we water plants? And so then you say, so you have to water a plant. People say yes. And I say, why? Because it'll die. You go, why? Right? That's all science. We're just a bunch of toddlers. We're just highly educated toddlers is all we are. We need to walk around, why? Okay, so what does the water provide?
High energy electrons.
Oh, I wish. That would be awesome. Yeah, low energy electrons. If water provided high energy electrons, water would be food. That would be awesome. Right? Okay, so water is a low energy electron source, or water is a source of low energy electrons. Fine. All right. So what does the plant then do with those low energy electrons?
Low energy electrons are excited.
Yeah, they get them excited into He in PS2. And the electrons from PS2, right, from photosystem 2, ultimately are used to produce what?
ATP.
Nice. ATP production. Very nice. But now they're low again. Bummer. Which is why there's two photosystems. So these low energy electrons are then re-excited into high energy electrons in PS1 and bonded to
NADP.
NADP, question mark, and the associated proton to make NADPH, because technically what happens is there's two electrons that are excited at one time. One electron bonds to the NADPH positive, making it neutral. The other electron bonds to the proton, making that hydrogen. You then bond your hydrogen to your your NADP, and you make NADPH. So what happens here is one electron bonds here, and one electron bonds here, if you really want to know. Thus, your high energy electrons sit here. Then, of course, NADPH goes, and the ATP go into the Calvin cycle. And ultimately that makes E3P, which can be used for two functions, purposi, right? That could be large amounts of ATP production or macromolecule production. There's your why. Okay, so why do you water plants? Because water is a low energy electron source. Why is that important? Because the plant excites those electrons and makes ATP for the Calvin cycle. The plant then re-excites those electrons and makes NADPH. The ATP and NADPH then go to the Calvin cycle for the production of ATP. Any questions on why we water plants?
No, not too much.
So next time, you have a potted plant, right, in the backyard, on the front porch, on the stairway, right? You're watering it. You'll be thinking, low energy electrons. Make your G3P go. All right, now let's take a look at core. Okay, this is the core. This is the only core that we do backwards, and there's a reason why. Because this core is ridiculously loaded with vocabulary, and if you jump into the core without a background, it's impossible. So we have to talk about all the handouts. We have to talk about all the processes, and then we work backwards. So it says create a simple diagram illustrating how the light-dependent reactions take place. Simple. That's image 10.5. Then talk about NADPH, right, which is simply the reduced form of NADP. So when you reduce NADP, that makes NADPH. When you do this in your core, combine these two. To have the diagrams, then the explanation separate. That makes no sense. Put them together. Okay, so put 1A and 1B together. Then discuss the role of water in photolysis. Done. Then it starts talking about the high energy electrons of photosystem 2. Those are images 1013 and 1017. Then it says diagram and discuss the light independent reactions. So be sensitive here. Be sensitive that these are light dependent, and be sensitive that these are light independent. Very, very different processes, of course. So down here, we're in the Calvin cycle. So just like earlier with the light dependent, how they said diagrams and explanation, it's the same format here. Diagrams, 1018, and explanation. So group 1E and 1F together also. And they drop this, this fancy phrase here, metabolic precursor. Metabolism is all of the reactions in a cell, all right? Whether those reactions are anabolic, bonding items together, or catabolic, ripping them apart. Anabolism and catabolism as a group is a collection of reactions we call metabolism. Okay, so metabolism is made of anabolism and catabolism. Fine. So metabolic means you're breaking or producing molecules. Precursor. This is something that comes before everything else. G3P comes before everything else. G3P comes before amino acids, comes before lipids, comes before carbohydrates, comes before nucleotides. Your G3P is what you start with, well, what the plant starts with, right? And then makes everything from that. So we refer to that original molecule as a precursor that you build with. Thus it's a metabolic precursor. Fine. And then G and H are separate. G is 1021 and 103, and then 1H is 109. So the overwhelming majority of these are driven by images, of course, and I would group together the images and the explanations, 1A and 1B, and then 1E and 1F. Any questions on that?
No.
No? Okay. Okay. So then let's do an animation and call it a day. Oh, animation should be down here. There we go. Plant, check it out. Okay. Apple, leaf, neat. Okay, so now we're going to zoom in on the leaf. So here's the surface of the leaf. Okay, I dropped the word stomata. All right, stomata are individual holes in the opening, or they are openings in the surface of the leaf. Okay, so that upper layer of a leaf has little holes, and those holes open and close. Individually, they're called stomates, and as a collection, they're called stomata. And carbon dioxide, water, and oxygen all go through these little holes. So here's good old carbon dioxide, right? CO2. And so it's entering through the stomata. So we're entering the leaf. Boom. So this is the inside of the leaf. Technically, this area is called the spongy mesophyll. Okay, nice, right? And then these are individual plant cells, okay? So now we're going to go into the individual plant cell. Now we're inside of the plant cell, and you go, whoa, what is all of this? I love this animation because this is really what the inside of cells look like. If I say the term cytoskeleton, does that ring a bell? Right? That's the structure inside of the cell that we never draw. Okay? And yet, the cell, the structure of the cell, these are just like the wood studs that are in the walls of our homes. These are just like the iron I-beams that hold up skyscrapers, right? This is the skeleton of the cell, and yet we never draw it. Why? Because then all your drawings look like this, okay? But this animation does a fantastic job of reminding us, hey, don't forget, the inside of the cell is actually very dense with this forest of cytoskeleton and fibers that support the structure of the cell. It's one of the few animations that I've seen that actually include that. So we are now then, they take the cytoskeleton away and we go, we can see Okay, so what are we looking at here? They show you the mitochondrion here. They show you a vesicle over here. They hint, here's a strand of the cytoskeleton just as kind of a reminder that it's still there. And then here's the chloroplast with the individual thylakoids on the inside, and then the thylakoids stack up into grana. Cool. So there's the CO2, right, diffusing into the chloroplast, okay, with the yellow rain of ATP. It's just so cool. And then here's all this chemistry that's going to happen. So we're inside of the chloroplast. We're in the stroma. They're showing you the 6-carbon molecule here. They've already made a glucose. And then the white and the red, that's water. Okay. In chemistry models, what they call ball and stick models, carbon is always black, oxygen is always red, and hydrogen is always white. It's an industry standard. Okay. Boom. So we're now going to go into the thylakoid. Okay, so there's the surface of the thylakoid. As if you're hovering above it, you're looking down at the thylakoid membrane. Right? Lots of things going on. That's the surface of the thylakoid. Now they've gone, and this is the side picture of the thylakoid. Where's that handout? I'm about to hold a handout up to the camera. I'm gonna go old school. So what they're showing you here is this. Okay, they're showing you just this part of it here. Now, what they've done is they've taken the ATP synthase from down here and they've moved it up here. Fine. Okay, so this is photosystem 2. This is your electron transport chain. This is photosystem 1, and this is NADP reductase. So these are your light-dependent reactions. So here's light coming in, little packets of energy, right? You can see the electron transporting here, right? You can see some of the protons here are sliding through. You're building up your proton gradient. This is the kind of animation where you'll need to watch this 3, 4, 5, 6 dozen times and focus on different parts of the animation. So now they're going to zoom in for us, okay? So water's coming in the bottom. Here's water, looks like Mickey Mouse, right? Water is oxidized during the process of photolysis. The protons are these little plus spheres here. The electrons are the little minus here. And then you will see the oxygen in red diffuse away. So water comes in, is broken apart, electrons, protons. Water broken apart, electrons, protons. There's oxygen leaving. Bye. Now, the electrons are excited, and here's the electron transport chain. Excitation, transport. And did you catch the pumping of the protons there? There'll be a proton here, proton, pump. Protons come in here and pump. There's the pumping of your protons. These are now low energy electrons. This animation goes so quick, it's awesome. High energy electron transported, low energy electron. Okay, the electrons then get re-excited. The high energy electrons are then bonded to NADP, the little red box, to make NADPH. Here comes NADP, NADPH. So we now have NADPH here. We now have all these protons in here. You have a proton gradient. These protons are then going to flow from inside the thylakoid out into the stroma. This is ATP synthase. Okay, so this is the inner thylakoid space. This is the stroma. And so the protons are going to flow from an area of high concentration to an area of low concentration. As the protons flow through ATP synthase by chemiosmosis, you will see the head of the ATP synthase open and close, open and close. There's 3 different lobes to this protein. And when it opens, it brings in ADP and P. When it closes and then ratchets, it forces the phosphate and the ADP together. And then when it reopens, it releases ATP. It's a highly physical process. Come back here. There's your flow of protons, and there's your production of ATP. And there it is from above. So there is a lot going on when they show this to you, okay? So I'll post this in the Facebook group now. I would sit back and I would watch this. Take it all in the first time, then go, okay, then back it up and watch just a part of it and watch that repeatedly. And then go to the electron transport chain and watch that repeatedly. And then photosystem 2, and then watch that repeatedly. And then ATP synthase and watch that, okay? Don't try to swallow this thing all at once. Take it in chunks. Then when we get together tomorrow, we will talk about the light-independent reactions and go through this animation again. And then we'll have presentations starting on Monday.